Hi friends, good afternoon. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about prokaryotes. Organisms can be classified into two different groups on the basis of their subcellular structure. They are prokaryotes and eukaryotes. Prokaryotes are the organism without a well-defined nucleus and other membrane-bound organelles, whereas eukaryotes have a well-defined nucleus and other membrane-bound organelles. Both prokaryotes and eukaryotes contain genetic material, but in eukaryotes the genetic material is found inside nucleus, whereas in prokaryotes the genetic material is found in a specific area inside the cell known as nucleoid. So let us discuss about prokaryotes. Prokaryotes are the single-celled organism or unicellular organism that lacks a nucleus and other membrane-bound organelles. The word prokaryote is derived from two Greek words. Pro means before, karyon means kernel. The oldest known fossilized prokaryotes were laid down approximately 3.5 billion years ago, that is, only about 1 billion years after the formation of Earth crust. Based upon molecular analysis, prokaryotes are divided into two domains. First is bacteria, second is archaea. Bacteria were formerly known as eubacteria and archaea were formerly known as archaebacteria. Some prokaryotes, such as cyanobacteria, may form large colonies, whereas others, such as myxobacteria, have multicellular stages in their life cycle. All prokaryotes are asexual. They reproduce without fusion of gametes. In prokaryotes, the main method of reproduction is through binary fission. In prokaryotes, horizontal gene transfer takes place through transformation, conjugation, and transduction. What is transformation? Transformation is a process of horizontal gene transfer in bacteria in which some bacteria take up foreign DNA from their environment and incorporate into their genetic material. Second is conjugation. Conjugation is a process of genetic recombination that occur in bacteria. In this process, there is transfer of DNA from one bacterium to another bacterium through direct contact. Third is transduction. Transduction is also a process of genetic recombination through which horizontal gene transfer also takes place in bacteria, and transduction is the phase-mediated transfer of DNA from one bacterium to another bacterium. Prokaryotic cells have various shapes. The four basic shapes of bacteria are cocci, bacilli, spirilla, and vibrio. First is cocci. Cocci are the bacteria that are either spherical or ovoid, whereas bacilli are the bacteria that are rod-shaped, and spirilla are the bacteria that are like twisted rods, and vibrio, they look like comma. In most of the prokaryotes, there is a cell wall that is present just outside the cell membrane. In bacteria, the cell wall is composed of peptidoglycan. Peptidoglycan is a major structural polymer that consists of glucan chain of repeating N-acetyl D-glucosamine and N-acetyluramic acid cross-linked by peptide chains. On the basis of the composition of cell wall, bacteria can be divided into two different groups. One is gram-positive bacteria, other is gram-negative bacteria. Gram-positive bacteria possess a thick cell wall that contain many layers of peptidoglycan and teichoic acid. In gram-negative bacteria, the cell wall is relatively thin that consists of a few layers of peptidoglycan and surrounded by an additional lipid membrane that is composed of lipopolysaccharide and lipoproteins. So this is the structure of a bacterial cell, and this blue layer is the cell wall. This blue layer is the cell wall. In some bacteria, cell wall is absent. For example, in Mycoplasma. Mycoplasma is a genus of bacteria in which cell wall is absent. Most bacteria have the gram-negative cell wall. The cell wall of archaea are unusual. Structurally, archaea are most similar to gram-positive bacteria. Cell wall is absent in some archaea like thermoplasma and ferroplasma. In archaea, 4 types of cell wall are found. In some archaebacteria, for example in methanogens, the cell wall is composed of pseudopeptidoglycan, whereas in some other archaebacteria, the cell wall consists of a thick layer of polysaccharide which may be sulfated. Few archaebacteria also have a cell wall that consists of glycoprotein, whereas in other archaebacteria, the cell wall consists of only surface layer proteins known as S layer. In addition to cell wall, the prokaryotes possess the following structures. They are glycocalyx, cell membrane, cytoplasm, nucleoid, genetic material, ribosomes, plasmids, flagella, pili or fimbriae, cytoskeleton, nutrient storage structure, intracellular membrane, inclusions, gas vacuoles, and microcompartments like carboxysome and magnetosome. Out of these, some of the structure may remain absent in some bacteria. First of all, let us discuss about glycocalyx. Glycocalyx literally means sugar coat. It is a network of polysaccharide found outside the bacterial cell wall, and the glycocalyx can assume several forms. If the glycocalyx is well organized and relatively tightly associated with the cell wall, such type of glycocalyx is known as capsule. Most bacterial capsules are composed of polysaccharides. In some species, for example in Bacillus anthracis, the capsule is composed of poly-D-glutamic acid. Capsule protect the bacterial cell from phagocytosis. It can be the cause of various diseases, and capsule is found in both gram-positive and gram-negative bacteria. Next is slime layer. If the glycocalyx is unorganized, diffused, and loosely associated with the cell wall, so it can be easily removed. Such type of loosely associated glycocalyx is known as slime layer. In this diagram, you can see the presence of red layer. This red layer, it is the glycocalyx. Below glycocalyx there is cell wall. Below cell wall there is a cell membrane. Inside the bacterial cell, this is cytoplasm, which is a gel-like matrix. There is nucleoid, which contain the genetic material, and there are numerous ribosomes. In addition to this bacterial cell also possesses two flagella, which are locomotory appendages. In addition to this bacterial cell also has hair-like appendages known as pili or fimbriae. Now let us discuss about the cell membrane. Cell membrane is also known as cytoplasmic membrane or plasma membrane. In bacteria, the cell membrane is composed of a phospholipid bilayer and different types of proteins. The proteins present in the cell membrane can be divided into two different groups. One is peripheral membrane protein and other is integral membrane protein. In case of archaea, the cell membrane is different because the cell membrane of archaea is composed of glycerol ether lipid, whereas in eukaryotes and in bacteria, the cell membrane is composed of mainly glycerol ester lipids. In archaea, the lipid tails are long isoprenoid side chains with multiple side branches, sometimes with cyclopentane or cyclohexane ring. In some archaea, the lipid bilayer is replaced by a monolayer. Next is cytoplasm. Cytoplasm, which is present inside the cell, the cytoplasm is a gel-like matrix that is composed of water, enzymes, nutrients, waste, gases, various structures such as ribosome, chromosome and plasmids. Next, nucleoid. What is nucleoid? Nucleoid is an area in the prokaryotic cell where the chromosomal DNA is located. In this diagram, this area is the nucleoid. So nucleoid is an area in the prokaryotic cell in which the chromosomal DNA is located. In E. coli, there is a single circular double-stranded DNA molecule which is its chromosome. Some prokaryotes may contain as many as 4 linear or circular chromosomes. Prokaryotic chromosomes are much larger than the prokaryotic cell. So, in order to comfortably fit inside the cell, the chromosome is folded through negative supercoiling. The most abundant protein in the nucleoid is U protein that work with topoisomerase 1 to bind to DNA and introduce sharp bends in the chromosome, thereby generating a tension which is necessary for negative supercoiling. By this process, the prokaryotic chromosome is comfortably fit inside the prokaryotic cell. All prokaryotes are haploid. It means they contain a single copy of each of the genes. And the non-coding sequences in prokaryotes accounts for an average of 12% of the prokaryotic genome, whereas in case of eukaryote, the non-coding sequences accounts for about 98% of the genomic DNA. Prokaryotic chromosomes are organized into polycistronic operon, whereas in case of eukaryotes, it is monocistronic. Next is ribosomes. Prokaryotes contain 70s ribosome. The ribosomes are responsible for protein synthesis, and the 70s ribosome consists of two unequal subunits. Larger subunit is known as 50s and the smaller subunit is 30s. Ribosomes are found inside the cytoplasm in free form or in the form of polysome. Next is plasmids. Plasmids are autonomously replicating extrachromosomal DNA molecules which may be linear or circular. On the basis of function, plasmids can be divided into 5 different groups: fertility plasmid, resistance plasmid, core plasmid, degradative plasmid, and virulence plasmid. First is fertility plasmid. Fertility plasmids contain tragenes that allow genes to be transferred from one bacterium to another bacterium through conjugation. Second is resistance plasmid. Resistance plasmid contain antibiotic resistance gene. They provide antibiotic resistance to the bacteria that contain them. Third is core plasmids. Core plasmids contain genes that code for colicin or bacteriocin that kill other bacteria. Fourth one is degradative plasmid. Degradative plasmid break down unusual substance like toluene and salicylic acid. Last one is virulence plasmid. Virulence plasmid transform a prokaryote into a pathogen. Next is flagella. Many prokaryotes have hair-like locomotory appendages. Flagella provide motility. And the flagellum of archaea is known as archaeolum. In some bacteria, the flagella can also function as sensory organs. In spirochetes, flagella are hidden within the periplasmic space. Such type of flagella are known as periplasmic flagella. The bacterial flagellum is composed of protein subunits of flagellin. The bacterial flagellum consists of 3 parts: filament, hook, and basal body. A shaft runs between hook and a basal body through protein rings in the cytoplasmic membrane that act as bearings. In gram-negative bacteria, there are 4 rings, whereas in gram-positive bacteria, there are 2 rings. The number, arrangement, and distribution of flagella varies from bacterium to bacterium. Many prokaryotes possess hair-like appendages known as pili. Pili is plural, pilus is singular. A pilus is a hair-like appendage found on the surface of many bacteria and archaea. The term pilus and fimbria can be used interchangeably. Pili are antigenic, they are also fragile, therefore they are constantly replaced. On the basis of function, there are 2 types of pili. One is conjugative pili, second is fimbria. Conjugative pili is also known as sex pili. They allow the transfer of DNA between bacteria during the process of conjugation. For example, F. pilus of E. coli is a conjugative pili. Pili are primarily composed of oligomeric proteins known as pilin. Then fimbria. Fimbria is a short pilus that is used for attachment. Fimbria is used to attach the bacterium to a surface. So, they are called as attachment pilus. They are also made up of oligomeric protein known as pilin. Then cytoskeleton. Previously it was thought that cytoskeleton is absent in prokaryotes, but modern techniques reveal the presence of cytoskeleton also inside the prokaryotic cell. What is cytoskeleton? Cytoskeleton is the collective name for all structural filaments. The structural filaments that are present inside a cell are collectively known as cytoskeleton. These structural elements play important roles in cell division, protection, shape determination, and polarity determination. Prokaryotic cytoskeletal proteins are FTSZ, MREB, PARM, SOPA, crescentin, main CDE system, bactophilin, and CFPA. Next, intracellular membranes. Intracellular membranes are also present inside the prokaryotes in addition to the cytoplasmic membrane. Some microbes contain intracellular membrane that play various roles, and these intracellular membranes are present in various prokaryotes like phototrophs, nitrifying bacteria, and methane oxidizing bacteria. For example, in phototrophic bacteria, one can find the presence of chromatophores, and these chromatophores are the intracellular membranes which are used for photosynthesis. These chromatophores contain various photosynthetic pigments like bacteriochlorophylls and carotenoids. Next is nutrient storage structure. Most bacteria don't live in environments that contain large amount of nutrients at all times. So, to accommodate these transient levels of nutrients, many bacteria contain different nutrient storage structures. For example, some bacteria store excess carbon in the form of glycogen. Glycogen is a polysaccharide which consists of glucose. Some other microbes also store nitrates in vacuole. In addition to this, sulfur is also stored as elemental granule. Next is inclusions. Inclusions are the non-living components of cell. The most common inclusions found in various prokaryotic organisms are glycogen, lipid droplets, crystals, and pigments. Another inclusion is volutin granule. Volutin granules are cytoplasmic inclusions that contain complex inorganic polyphosphate. Volutin granule is also known as metachromatic granules. Next is gas vacuoles. Gas vacuoles are also present in many prokaryotes. These vacuoles are membrane-bound, spindle-shaped vesicles found in some planktonic bacteria and cyanobacteria. The main function of gas vacuole is to provide buoyancy. Next is microcompartments. Bacterial microcompartments are widespread membrane-bound organelles that are made of a protein shell that surrounds and encloses various enzymes. Examples of microcompartments are carboxysomes and magnetosomes. Carboxysomes are prokaryotic microcompartments that contain an enzyme Rubisco and Ca. Rubisco means ribulose bisphosphate carboxylase oxygenase and carbonic anhydrase. These carboxysomes are present in cyanobacteria where they help in photosynthesis. This Rubisco is the most important enzyme in the carbon dioxide fixation. Next is magnetosomes. Magnetosomes are the microcompartments that are found in magnetotactic bacteria that allow them to align themselves along a magnetic field. Friends, this is about prokaryotes. Please share, like and subscribe and comments on my video. Thank you one and all.